a lot of people have alluded to the fact that there's a lot to talk about with therapy this year, and I think Dr. Zuckner was worried that he was going to steal my thunder. There's a lot of thunder to go around, and frankly, that's one of the themes. One of the first slides that I'm going to talk about here is some of the important themes is the FDA willingness to approve innovative therapies, huge amounts of investment. So far this year, and I think this was as of even a couple of months ago, it was over $2.5 billion invested just this year so far in cell therapies. So you can see that this is literally an area that's on fire. Drugs are being approved that are helping in rare disease. And you saw somebody walking after being blind, and being able to see around an obstacle course. I mean, that's the definition of a rare disease. That disease has about 1,000 people in the world that are affected. So that's as big as PLS, right? I mean, we're talking about PLS being a very small portion, even rare in terms of rare disease. Well, that blindness indication that Dr. Zuckner showed yesterday is for the same size population in this world, and still they brought that drug to market. So that's really good news for all of us. Rare disease is not being ignored. It's still a difficult challenge, but it's not being ignored. Let me just sit back. I went forward a little bit too far. This is my title slide, and it's got some pretty amazing updates just on the front. What you'll see under FDA status is 4 major approved drugs. And sorry, I get a little bit choked up because some of these are a little bit personal. It's helping a lot of communities, right, like ours. And we're going to be on this list, but this did not exist last year. Last year, when I spoke to you at this conference, we had 1, and it is with an organization that I'm part of, that first one, SMA. I'm on the board of directors also for Cure SMA, and we didn't expect an approval at that time, but we got it. And there's been 3 other major approvals just this year, and there's a lot more that are ready to come. These are all in rare disease, and it's pretty amazing. Lots of different mechanisms. So you'll see the different kinds of cell therapies up there. I'm going to go through some of those today. And that's where I want you to ask questions. I want you to please, already a question.
Jump in, but give me a short synopsis of what gene therapy.
I will. I'm still not off my first slide yet, so I will go through that. There's lots of different kinds of cell therapy, but basically the question is, what is a gene therapy? What is a cell therapy? And it's pretty broad. There's lots of different ways to modify cells so that you can treat therapy, not symptomatically, but treat the actual root cause of the problem. That, in effect, is the way that I think about cell therapy. You're actually fixing the cells, either through the genes or through replacing cells, or some way to actually restore a disease state into more of a normal state. That in effect is what we're talking about. Any kind of therapy that does that. Okay? All right, so let me just move on here. So I'm going to talk a little bit today, right, about some of the themes, and I talked about that already a little bit, went to that slide, but basically this area is on fire, right? In some ways it doesn't feel fast enough because it may still be a bit of a distance from helping anybody that's in this room, but the work that we're going to see here is going to help progress for folks in this room. Even if we're not working on that today, and all the research that you've heard about so far leads to drug therapy, even if there aren't clinical trials today, this work will help this group, okay? I'll talk a little bit about phases of a clinical trial. I think you've got from experts better than I a little bit of a preview of that, but I do want to talk a little bit about what those phases are and what it means, because I do refer to phases, and I hate to refer to things that people don't know about. There is no mystery here. There's a lot you can understand, but there's a lot of detail, and I don't want to get too drugged down in the details, but I do want to show. Dr. Zuckner yesterday, I'm very grateful to him for talking a little bit about basic genetics, because I think that's important to happen early in a conference, because if you don't understand some of those words, it can be challenging through the rest of the day. But I do want to cover a little bit about basic genetics and really how we can deliver some of these cell therapies, just like John was just asking. You know, what is a cell therapy? It's how you get some of that genetic material to where it needs to go in a cell. And then I'm going to talk about some examples, and I may go into too much detail in some of these examples, so so I may cut it short, but examples in spinal muscular atrophy, in some of the cancers, in the blindness case, and so forth. But I want to leave plenty of time for questions. Okay, so again, I already went through these themes, but the FDA willingness to approve innovative therapies. I heard Scott Gottlieb in person in Chicago just a couple of weeks ago at the American Society for Clinical Oncology iterate things that I've heard him or read him talk about and write about in articles. I heard it from his mouth that this is a huge area for the FDA, right? They're looking to have innovative therapies be approved. And that's huge for us because you can imagine with all these new technologies, there are lots of questions. And I've highlighted those questions the last 2 years. But you don't want to have analysis paralysis and you don't want to wring your hands. You want to be able to do in a responsible way, take risks depending upon rewards. And so you'll see in some of these cases, especially some of the cancer therapies, are in some pretty extreme cases where there's a significant likelihood that you're not going to have a remission on its own. And so you can have interventional therapy. And that can help us learn and apply that method to other areas. But there's some pretty extreme cases. But the FDA is willing to take steps and move a little quicker. Again, investment, and that this is really helping rare disease so far. So I gave you that snippet of what's approved, but I'm going to keep coming back to this slide, and the slides are going to be available on the web, so don't feel like you have to write all this down. It's just a snippet. It's a beginning. It's a snapshot of some of the major work that's happening that is approved and some of the stuff that's relevant for us, okay? One of the things you'll see down there at the bottom, and if I use my pointer, it's only going to point on one screen, so I apologize. I'm pointing over on this side right now. But if you look down on the gene replacement side, the last from the bottom, there's a couple of people in this room that this affects directly. You see actually the SPF has funded an SPG15 large gene vector replacement, and there's a couple of folks right over here that you may have talked to, and you see their poster out in the hallway, where we're working on this in spastic paraplegia. We can't yet for PLS because we don't have a lot of forms of PLS, genetic forms of PLS that are identified, but it's ready for when those are identified as well. And so we do see some action in this area. It's still early. It's still in the preclinical phase. We're not close to approval yet, but it's being worked on and it's active, okay? So I'm going to go through some of these examples. It's a lot of words right now, but by the time we come out of this talk, you'll know a little bit better about each of these different line items. So let me go through a little bit about clinical trial structure. And this, it's actually quite fuzzy. I apologize for that. You've probably who's heard of phases of clinical trials? Who knows exactly what that means? A couple, but not many, right? Why do we have phases of clinical trials? This is all experiments in humans. And experiments in humans have to be done extremely carefully and thoughtfully, because there's been experiments in humans in the past, in times of war, that have not been ethical, that have not been appropriate, and even not in times of war. And some of that has guided our principles of how we do experiments in humans thoughtfully, ethically, and move forward with therapies, but not so fast that you're actually taking advantage of a population that needs the most help. You don't want to be taken advantage of, but we also want to move quickly, so it's a balance of those things. So when we're talking about a phase 1 trial, it's all about safety. You are testing a particular drug in a population that is not diseased to see is it safe. You might be varying the doses to know what that threshold of safety might be, but you're doing a proof of concept to say, is this drug tolerable? And you're doing all sorts of studies on that person to say what might be going wrong, because you don't want to put this into a population that's affected unless you know that it's safe. There's usually volunteers. Obviously, this is all a volunteer piece, but that phase 1, that's what it's all about. The only endpoint is safety. You're not trying to figure out if it's helping anybody. You just want to make sure it's not going to hurt, okay? And in rare disease, a phase 2 and a phase 3 might be combined. And it's because there's so darn few people in the population that it doesn't have the same meaning as it does, for instance, if you're going to be doing a new kind of like arthritis medication, where a lot of people have arthritis. And you might be able to do a phase 2 study where you're saying, okay, we're going to test a small population, and then we're going to test a much bigger population, maybe tens, maybe thousands, maybe tens of thousands, and in a phase 4, maybe really large numbers. And sometimes that's monitored after a drug is actually marketed, okay? But you're looking for an outcome measure. Here, the example is cardiac output, but maybe if it's something in our disease area, maybe it's a 6-minute walk test, right? How far, how fast can you walk in 6 minutes? Maybe it's a biomarker study. You've heard about a couple of different kinds of biomarkers. Maybe it's the MRI screen that Dr. Paganini was talking about, right? Maybe it's a changing of that biomarker that you would measure, something you would measure to show that there might be a change. But any of those outcomes are measured in those phases. And it's usually placebo-controlled, which can seem quite cruel, because it means that there is a group of maybe 20 patients, maybe it's 10 because we can't recruit 20, and a third of them might specifically, in a blinded way, not get drug. And the reason to do that is because you want to be able to compare those that get it with those that don't. But a lot of the trials that I've seen in this area, you might have a third placebo control instead of 50-50. It'd be better to have it 50-50, but to be compassionate to a group that needs a therapy, maybe it's 2/3 and a third. And after a year, maybe everyone switches over to the drug instead of complete all the way to the end. So that's a way that drug companies are working with us to have this be a little bit more compassionate to a rare disease community. But the most important thing is, if you don't have that data, you're not going to get coverage of the cost of the drug by payers, by health coverage, because we're seeing that. These drugs are going to be extremely expensive, and I think that you may have seen that on the previous slide. So if we don't do a good job showing the improvements, you're not going to be an effective treated community once the drugs come out. Does that make sense? Sorry, that's kind of dry, but that's something that we are seeing in some of the other areas that are being helped. And it is really important to understand, because otherwise it can feel like everything's really slow, but that's why it's important to do it well. Are there questions about that? Question?
Most of us, we may not get a huge improvement, but to the individual, it may be enough of a quality of life improvement to move forward. But the drug companies, from a financial perspective, may not do it. So how do you bridge that gap?
The question is one about outcomes. How do we measure actual improvement, right? You might feel better, have a self-reported outcome that's good. It's a very good question, because if we don't have good outcome measures that are at least semi-qualitative or quantitative in some way, and not just self-reported, yeah, I feel better today, or generally I feel better, or I don't feel better, you're not necessarily going to be able to have measured impact that the FDA can approve and that payers will pay for. So you can have a drug that works and nobody will pay for it, which is, I would say, worse than having no drug at all, right? To have something that's there. So we've got to have quantifiable outcome measures, and it's one of the reasons why SPF here funds that kind of research into outcome measures, either walking or clinical or a biomarker, something that you can measure in your body, that would be an appropriate measure. And we're still lacking those areas, but we're trying to get ready so that when it's time for us to start a clinical trial, we can have an outcome measure that we can follow. It's very important for this organization to fund that kind of work. Does that answer your question?
Yeah, thanks.
Other questions?
I'm wondering, I have HSP, as I told someone before, but anyway, I'm wondering if is it gene replacement that's really of interest to you?
Yes. The question is about gene replacement therapy. Let me go through some of the examples and see if you still have a question, if that's okay. I don't want to just lecture, but some of this stuff I will get to, so I'll get on to it now and hang on to that question and let me know if I answer it or don't. So before I get too much into gene replacement therapy, and which is some of the source of your question, let me just go a little bit into what Dr. Zuchner covered, which is We're talking about whole organisms here, right? Our human body. There are, I think that he said 40 trillion. That's probably a pretty good number of cells in our body. And of course, our disease is very few of those cells, but some of the problems may exist in other cells as well, and we just may not know what that phenotype is of those other cells. The really impactful ones are in motor neurons. But if you take each of those cells has something called a nucleus in it, and inside of that nucleus is genetic material in the form of chromosomes, right? If you take all of that DNA, or deoxyribonucleic acid, a little cheat sheet at the top here, and you take that DNA and you stretch it out, it's like 1.7 meters or so crammed into each and every one of these cells. Each of these cells are capable of making an entire person, right? It's amazing, right? That is the blueprint. That is the programming to be able to make cells. And if there's a problem in one of these genes, in one of these letters of this DNA, you can have disease. Not necessarily every one of them. Yesterday, Dr. Zucker talked a lot about variation that's normal. Just because there's variation doesn't mean that that's bad, but some of that variation can cause disease, and in fact does, okay? So a little bit about these letters as we talk about it, because it's all about the nucleotides, or the letters. These are the basic building blocks of DNA: A's, T's, G's, and C's. If you've got kids, or if you've got grandkids, they're doing this now in high school, right? I did some of that in high school, but most of it was in college and in my postdoc days and my doctoral days. But they're teaching this earlier and earlier. So this is becoming more common knowledge and fundamental. If you have questions, you can always review the videotape, and you can ask some other people in your family or close friends. So G's, A's, T's, and C's, it's a code. It's a code in a string, just like any other code, and we learn how to read it the same way that you'll learn how to read a book. It's a different language, but it's the exact same thing. We know how to decode it. There's plenty that we don't know about the book. We're still learning more about it, but we know how to read the fundamentals of this code, okay? DNA is an incredibly robust and stable structure. You saw Jurassic Park where DNA was fossilized? Literally, it is stable for, in some cases, tens of thousands of years. It's a very stable molecule. So inside of each of our cells is a very stable blueprint that passes on from generation to generation. There can be changes in DNA. So even though it's stable, doesn't mean that it doesn't change. And that's part of what makes us all look different, be different. It's part of what's amazing about human beings and all life on Earth. But a deletion in DNA can be an entire area that's missing. And here it's this red X, a deleted area. Do you think that might cause a problem? If you're missing a big chunk of your DNA? Absolutely it could, right? Maybe not. Depends on how big it is. But certainly that can cause a problem. If you have too much DNA, you think that might be a problem? Could be, right? Too much is not necessarily a good thing. Having the right balance is important. What about an inversion, where maybe you're supposed to have a section that reads, again, like a book, maybe chapter 5 through 6 are reversed? Do you think that might be a problem? Could be, right? Maybe not. Not everything is going to be a problem there, but in this case, maybe it is. And you can have single gene or single nucleotide changes. Do you think that changing the letters in a word can make a difference? Maybe, right? And maybe that's something that we need to fix. So if you have questions, let me know about that, but I'm going to be moving on. That's as much as I'll touch on here, but I'll come back to it as we talk about how to correct some of these spelling errors, okay? So a little bit about cell therapy. This gets to your question, and if this doesn't answer your question, let me know. So what is cell therapy? What does it do? Altering the DNA sequence of an organism for purposes of treating disease is generally what's called gene therapy or cell therapies. All right? There's a few different ways to do it, and it's still a definition, honestly, that's being formed. I was calling it gene therapy before this, but really now it's called cell therapies. And I think I sent around a couple of books. Maybe 4 of them that are around. And that's a magazine, a publication that is reviewed by professionals and people publishing it all the time. And it's called gene therapy, but now often they're called cell therapies, all of these kind of general, so that nomenclature even is changing. But an important challenge of gene therapy is targeting the correct cells and enough cells to help with disease. You can imagine to get therapies into the right place in the brain, pretty darn challenging, right? But we've shown that we've been able to do it in some cases. We didn't know that 5 years ago. 5 years has made a world of difference there. We don't know, I don't know in 5 years what it's going to look like. We were talking last night some about, what does the future hold, right? How is everything going to look? In some cases, we have no idea. Some of the things have gone faster and better than I ever could have hoped for, and some things are slower, right? But being able to target the right cells is a really important problem for cell therapy. Getting cells there or getting therapy too. But we've been able to solve some of those problems, at least in some cases, okay? Therapeutic genetic material can be delivered to cells via, big word here, sorry, adenoviral vector or adeno-associated virus. I always call it AAV. There's a whole bunch of different what's called serotypes. AAV9 is pretty popular these days. There's an AAV10, and there's a couple of companies I'm going to talk about later that are making whole suites of these vectors. Now these, you would think that a virus is a bad thing, but this is a virus, and I'll talk about it soon, that it doesn't incorporate into your DNA. It can just deliver a payload of genetic material. Think of it as like FedEx. It's a FedEx truck that's getting the packages to the spot where it's got to go, all right? And it can hold a lot. It can hold a lot of packages. It's not infinite number, but it's pretty good size. Now, if you're trying to deliver something that's small, little lipid nanoparticles injected just intravenously into your blood system can work in some cases, and that's gene replacement therapy that's happening today in clinical trials, all right? So if it's something that's little, you can do it through a little liquid lipid nanoparticle. If it's something that's bigger, you might have to package it in a virus and have it be selectively delivered to cells, okay? There's something called CRISPR. CRISPR is I've talked about this before in previous years, although almost everyone is new. Amazing advances, right? We were talking about CRISPR as kind of aspirational 2 years ago. I've even included a couple of slides from a few years ago because it's almost funny how far we've come and how we were thinking, wow, this could be really great. And 2 days ago, we got news from Sarepta that, one of these studies that we talked about 2 years ago is looking great. So I'll talk about that some. CRISPR needs to be delivered via an AAV process because it's quite large. But basically, it's a natural biological process that we've discovered in the last 20, 25 years. And bacteria actually use this to protect themselves from other host agents, in this case, bacteriophages. And we've been able to realize, oh, we can use this to actually incorporate new genetic material into cells. And so CRISPR contains a guide RNA, a specific sequence that allows you to target to the right region, again, the right part of the book that we're trying to read, and cuts out a piece and can allow a change of precise, what you might call an edit of a letter to a letter that isn't going to cause disease. All right, so we'll talk some about that. That's in this way, it's been used as a research tool for a long time, and going to be used as a therapy tool by editing specific DNA sequences. That's one mechanism of cell therapy. So a little bit about this AAV or lentiviral vector mechanism I told you about, because I don't want you to be scared of the virus piece, right? There are some viruses that can cause some pretty awful things. This is an example where it's a kind of virus that transfects both dividing and non-dividing cells. So you don't want to just incorporate into cells that are growing, or else it'll go into like your digestive gut and maybe your blood cells, but not into your brain cells that maybe aren't dividing all that rapidly, right? So this is an equal opportunity infector. It'll infect different kinds of cells, and that's a good thing in this case. It doesn't integrate into your host genome. Why do you think that might be important? What's that? Doesn't alter your DNA, right? So unless you want it to actually incorporate, it's not going to integrate and screw up something that's already working well. You don't want to cause more problems. And with this, we always worry about off-target issues, things that you don't intend. In fact, that's caused a lot of people a lot of work and a lot of pause. And it's one of the major safety issues being investigated by companies and the FDA and so forth. So you don't want to screw up what's working well. That's really important. So that's a feature here. It's easy to produce at a high titer, so that's going to be important for manufacturing and quality control of manufacturing, which sounds ridiculous now, but when you're at that stage, and if you can't make the drug, that's not ridiculous at all, right? You've got to be able to produce this efficiently and at a high titer. And we now have in the world, this is why I say these drugs may not be impacting this community now, but these manufacturing facilities are starting to be developed around the world. Some of them in airports, because you've got to get cells and be able to make it fast enough and get it shipped out. That point, that's really important where that's being developed. So that capability is being developed, and it may not be for our therapies today, but it will be in the next 10 years, right? Okay, let me just move on. So it can be used for in vivo gene delivery. So you can fill, if you look at this structure, right, this ball of blue that's up there, if you can see it well, that is a ball. It's literally hollow and holds a payload. That's why I call it a FedEx truck. It literally holds material inside of it, binds a cell, and puts that genetic payload inside. That's what viruses do. That's what this virus does. We're using it for that purpose. Make sense? All right, little FedEx truck. All right, this is a company, and I've got the logos. I've got logos of lots of companies up here because they're companies that I don't have any vested interest in them, but I'm working with them in a lot of cases, or I know the folks and I highly respect them. Homology is a company up in Cambridge that the chief scientific officer is a good friend of mine, and they're making a whole suite of adenoviral vectors. And it's going to be used, I think, as a platform for the future to make individual gene therapies easier to accomplish, easier to get through clinical trials, manufacturing, reimbursement, and delivery. That's what I'm hoping. We need to change to have that be kind of a rapid model. It's not today, but it's still early days. And I think companies like Homology that are investing a lot of money into this area to develop a suite of these FedEx trucks is going to be really key. Some of them might specifically find liver. Maybe some specifically find, something in the central nervous system. It depends. You also don't want to have an allergic reaction to these vectors because then you'll shut down how effective they are. So right now, about 10% of the population can't receive some of these treatments because your immune system attacks it as foreign and clears it out, which sounds awesome, usually for viruses, right? You want to be able to fight off your viruses, but you don't want to fight off the virus that's delivering your essential therapy. So it's kind of this funny balance. So they're making a whole suite where people do not have an allergic reaction or do not get rid of or clear or have sensitivity to that suite of viruses. Does that make sense? Okay, so I like what Homology has on their website. They're talking about a couple of major ways that their viruses can be delivered: gene editing and gene therapy. Editing, I just talked about a little bit with CRISPR, right? Where you're changing a couple of letters. Gene therapy, in this case, their model, you can see kind of in their little picture here, and I'll point to it on both screens. They're calling it an episome, and it's right here, over here. Instead of integrating into your chromosomes, and like you were saying earlier, screwing up what exists already, maybe it interrupts something that's important, it'll just survive inside of a cell stably and make its gene product and fix what's wrong. That's the concept there. So they have two kind of major mechanisms of action. Questions about that? That one's pretty important, right? So you can actually fix what's broken, or make a little non-chromosomal episome. And in some cases, you can actually have homologous regions on each side and actually correct the gene that is incorrect. But that's not necessarily what Homology is looking to do here, but that can happen as well. So these are fundamental tools that we're going to be talking about in all the examples. Any questions? Okay, cool. That's good. You guys are doing great. So why does cell therapy matter? Maybe to this audience that's obvious, but editing DNA sequencing and DNA sequences in a cell is an important capability for both research and therapy. Again, we've been using this in research for years to use cell models and mouse models and so forth to say, if we take this gene out, what happens? Can we make a model of disease and then treat a mouse or cells with drugs to see if that reverses any phenotype before you test it in a person? People have been doing that for years. I can guarantee that a lot of the researchers in this room do that routinely in their lab. They've been using this technology for dozens of years. Not necessarily in people though, right? So we're making that leap to people. For research, CRISPR-Cas9 can create cells. I just kind of explained that. For therapy, CRISPR can be used to edit DNA sequences for a particular benefit. The first CRISPR clinical trial was announced literally 2 years ago when we were in conference. And in that case, it was for Duchenne muscular dystrophy, and there was also one for cancer. And both of those now have had progress, and one of them's been approved. So it's pretty darn cool to see that progress. So let's see. So we first talked 2 years ago at this conference. Like I said, we had gotten the green light. The procedure targets bone marrow cells that are removed from the body. So in the case of cancer, if you have a particular form of cancer in your blood cells, the nice thing about targeting, remember I said that it's kind of hard to target the cells? You can actually take bone marrow out. Pretty painful. You have to actually go in and get your bone marrow, so that's a little bit painful. But you can take out bone marrow, and you can actually infect those cells in a dish and then put them back in. That's pretty cool. So that's why that progress in those kinds of cancers have moved so darn quickly. It's because there isn't a targeting problem. You can and a lot of those cancers can be quite severe and significant. So it's a population that really needs the help quickly, okay? So that's one reason why we're able to move quickly on those. And this first one, it just happened to be at the time. I mean, this is an old slide. I wanted to include it because it's funny how fast things move. This was funded completely independently by the former Facebook president, Sean Parker, who literally wanted to invest in immuno-oncology, knew that there was promise, and wanted to propel this forward. Dollars make all the difference in the world here. $250 million is an awful lot. I mean, you can do a lot with a lot less than that. But in this case, it was completely philanthropic, and he got it done, right? And that is a therapy that I'll be talking about here in a minute that has been successful. So what are some of the risks? I do want to, I'm up here talking very passionately about some therapies that can really help, but I also want to be quite clear about risks, right? So you can have off-editing or off-target modifications, but I'll tell you, it seems to be a less of a concern than it was even not too long ago. So far so good, but it's something we really need to keep an eye on. It's very important that you know that CRISPR treatment of adults and children is not for editing germline. Who knows what I mean by germline? Germline means eggs and sperm. It's not going to impact future generations. We're talking about editing cells that are part of your body that are not egg and sperm. That means you still have risk of passing this on to future generations. Editing of eggs and sperm is a whole different area, and that's not what we're looking to do. That's not even considered ethical at this point, right? It's something that's maybe a hot topic in the news and things that people are concerned about, like sci-fi. We're talking about editing all of the cells, but not egg and sperm, right? So maybe in your central nervous system. So there is still risk that you can pass this condition on to children. Does that make sense? Okay, good. CRISPR is personalized medicine to an extreme. I mean, we're trying to fix something that's specific maybe in you, and maybe only you in the world, or in 1,000 people that have blindness, or 1,000 people that have PLS, or that have a particular typo in their SPG4 or spastin or elastin or REAP1, whatever your gene is, it's about fixing that piece, right? It's personalized medicine. You hear a lot about that. And I put at the end here something that I'm hoping for. Maybe this is what homology can help with, but we have to be able to get to a point where Different gene therapies that are going to be so personalized don't need to wait a dozen years to go through an FDA clinical trial, but we're not quite there yet. But we need to be able to change that paradigm so that it's still safe and it's still appropriate, but not necessarily going through an entire clinical trial for every single personalized therapy. Does that make sense? Platform approvals. And we need to get there. There's work to do there. Okay, so back to the list. How am I doing for time? Well,
10 or so minutes or so. You're doing great.
so I'm like a quarter of the way through and I've got 10 minutes left. Okay, we're on track. Let me go through this really quickly because I want questions and answers, okay? So I was gonna talk some about this top line first and really kind of go through each one individually, but I'm gonna have to zip through. What you'll see on the end, it's amazing that these things are improved. You're gonna see some eye-popping costs, and I don't wanna shy away from those. But it's important that you know that one of the major reasons patient advocacy groups are important is to be able to have your back for things like this, right? One of the major things that we're doing now as part of Cure SMA is to make sure that the drug is accessible to people that need it by lobbying through government agencies, NIH, Health and Human Services, all of those things that matter. We have, you shift from, and we're still funding research in that case, and I think in this organization we'll always fund research, but you also shift to other kinds of services and other ways that we need to support your community. This organization is going to be even more important through that phase. Does that make sense? And even coming out of it. Because these numbers are not sustainable, right? But if it's covered well by insurance companies and there's good evidence for it, we're going to be able to get access to patients that need it. Period. That's the goal. It must happen. And that's why we're doing what we're doing. So I'll talk a little bit about SMA. I'll talk a little bit about the two different CAR-Ts and what they mean. So first, Spinraza, it's an ASO. So this is an antisense oligo. This is one of those little tiny sequences that's delivered by lipid nanoparticles. In this case, it's delivered via an injection intrathecally. That means it's actually injected into the lower part of your back so that it can bathe the brain and the spinal column in therapy, not your entire body, right? So it's harder to deliver, it's expensive, but it was the first in its class, and that's how they wanted to make sure that it was getting to where it needed to go. Follow-on therapies are going to be, in some cases, there's pills that people can pop. In some cases, it's injections intravenously. So it doesn't mean we're going to have it intrathecally, right? I don't want to worry anybody about that. But in this case, that's how it's delivered, okay? Spinal muscular atrophy, it's a motor neuron disease just like us. It's a motor neuron disease that has a therapy today. That's important for everyone to understand. It's different kinds of motor neurons, but they're still motor neurons, okay? SMA in this case is quite severe. #1 genetic killer of kids under the age of 2. These kids are not dying at the same rate anymore. That's important. Carrier frequency is incredibly high. In this room, there are probably 4 or 5 carriers for SMA and you don't know it. If you happen to have children with somebody that's also an SMA carrier, you have a 25% that your children will die before the age of 2, and it'll come out of the blue. You didn't necessarily see it in your family, right? It's just tragic, absolutely tragic. The good news, post-treatment updates are being presented by some of the sites that are treating, and we had data before approval in children. We did not have data in adults. And I won't have time to go through it. SMA has lots of different severities. A lot of kids die before the age of 2, but there are adults that have the condition. It's a whole spectrum. Adults were not part of the clinical trial, but they were approved by the FDA to be treated. So nobody knew what was going to happen when we put this therapy in an adult. You don't want to hope for the best. You want to be realistic. And we got, last weekend in Dallas, updates for the first time of adults that are being treated, and it's incredibly positive. People are regaining function, and that's, I think, more than anybody could have hoped for. That's pretty amazing. You know, you hear about patients that literally need help sitting up, right? And that can be a risk of leaving somebody alone. So it's on a caretaker, right? Or a service animal to help prop back up. But literally, kids or adults are regaining function to be able to sit up and even to be able to comb one's own hair or to be able to lift your arm up. I mean, there's little milestones for individuals that can be quite impactful to their day-to-day, all right? All the way down to kids that are zipping around in wheelchairs that previously would be dead. Pretty amazing. So, hard to overstate that. Okay, I will not have time to go through all of this, what is SMA, but if you have questions, let me know. The genetics of it, I'd love to go through it. I will focus on one little piece here in the genetics of SMA, and it's what does an ASO mean? It's an antisense oligo, and it was discovered, it happens to be discovered in a place where I did my postdoc. It wasn't in the same lab where I did my postdoc, but UMass Medical in Worcester, Mass. Designed this little molecule, and I'm pointing to it, I'll point to it on the other side, called ISSN1, and now it's called Spinraza. It's a little antisense oligo. So what I mean by that is it's a short sequence of letters that bind specifically and inhibits a splice repressor that causes disease. And it's more complicated than that, but say that it blocks a repressor. And putting that into the right therapeutic target at the right time can allow for more expression of a protein that you need, or else your motor neurons will die, okay? So it blocks that and prevents death of those motor neurons via mechanisms, quite frankly. We don't understand all the mechanisms why. We're still doing basic research in that area. Make sense? Questions? Okay. Pretty effective. And because I've talked about this before, and I was going to go through modes of inheritance and I didn't get the chance to, because this is a recessive gene, how many copies of a healthy gene do you need to be healthy? I'm seeing the right answer. I'm seeing one. The answer is one. If you have one copy, you don't have to have two good copies. You don't have to have a good copy from mom and dad. You just need a little bit of this stuff and you're okay. So that may be in our favor that this is a recessive condition, because you don't need much protection to get what you need biologically. In some cases, you've got a dominant condition where just one bad copy can actually cause disease, and maybe you need a little bit more in those cases. But I don't want to get into that too much. If you have questions about it, let me know, because I didn't go through inheritance patterns. Okay, a lot on genetics of SMA, and lots of therapeutic approaches, some of which may be relevant for us because muscle neuroprotection might be important, and certainly muscle protection might be important for our patient population. There's a significant drug pipeline because there's only one gene that causes like 95% of SMA, which is very different from our population where it's dozens, even in some cases hundreds of genes, right? So it's attractive because there's one therapeutic target. It's one reason why it can go pretty quick, but that's going to pave the way for a lot of work that we can follow on, okay? So a very active drug pipeline. This is not a cure for SMA, but disease progression is often halted and even reversed. First adult data are quite positive. Cure SMA is an advocacy group that's working with payers, government lobbying. Cure SMA is also working on RUSP approval of SMA for carrier testing, and we're also working state by state to make sure that testing is done as early as possible. And folks like Invitae that are actually here are doing a lot of that important testing around, especially in the United States, but even around the world. And it's really good work. It's going to catch kids before they get sick. And that's a really great therapeutic window. You don't want to have to wait till an adult to actually reverse symptoms. It'd be good to catch them early. All right, so that's that one. Now, we've literally covered one example. I probably got like 1 minute left. So again, I'm going to zip through. All right, so you probably heard big news about something called CAR-T. CAR-T is chimeric. It doesn't matter. I've got it in one of the previous cells. It's a cell therapy where you're taking out bone marrow. You're actually doing CRISPR, 3 different kinds of CRISPR on it to make it attack cancer cells. And you put it back in the body. It's like phenomenal. You're making little heat-seeking missiles, and you're putting it back in your body, you grow it up. They literally, in a course of 3 weeks, take out bone marrow, do specific gene modifications via CRISPR, and then they grow it to be billions of cells, and there's movies on it. You just look at it on YouTube, type in any of these therapy names, you're gonna see movies on it, put it back in, it literally heat-seeking missiles your cancer cells. They've got cancer cells, in some cases, have particular markers on them. They're rare, but it's working. It's curing. You've got an 83% remission rate out of the 83 kids, in this case, that were tested that previously had failed therapies were put in, they would definitely die. 83%, I think it was 67 of them, had remission. Amazing? It's amazing, right? It's a whole new world. It's a whole new world for this entire population. That's just, there's going to be more examples. So Novartis has said that if patients don't respond to therapy, the company will not charge for the drug. That's pretty cool, right? Because it's such a high positive rate and success rate, this company is willing to take on some of the risk and say, you don't have to pay for the therapy if it doesn't work. That goes part of the way toward balancing cost, right? But one of the things that this article is mentioning is the cost of the drug is not the only cost of this. There's delivery, there's hospitalization. You can have adverse effects. In fact, sometimes when you have an adverse effect here, a cytochrome storm, or cytokine storm, that can mean that it's working really well, but you've got a fever that you need to manage. I mean, you're messing around with somebody's immune system here. You can imagine all the things that can go haywire, but that can be good, right? So that can be pretty expensive and cost money. But things are happening, and things are happening to address cost. Please ask a question.
With these augmented cells, are they actually going back into bone marrow so that they can use treatment against this cancer, or is the actual process of doing CRISPR on the cells so that they will actually die off and not become?
In this case, they're specifically selecting for T cells, not necessarily progenitor cells. So some progenitor cells may be impacted, and that would cause, but we're not selecting for stem cells. We're selecting specifically for T cells and modifying those and growing those. But at the same time, yeah, I mean, these are cells that are dividing and growing, and that's one of the things that can cause concern to say, look, are we putting something in that's not necessarily going to be good in the long term? And we'll see. You know, right now, it's a lot better to have a treatment than to have no treatment, but we're still watchfully waiting to see, would this be applicable in areas where maybe it's not a terminal condition, and you might not want some of the undesirable side effects later, and we're just waitfully watching. And others may have even better information than me on that. I mean, please, you know, there's other experts in the room here. Question?
If CRISPR doesn't affect sperm or egg, does that mean that it can be used? In other words, family could not be as worried about passing it on to their children, and could the children then be treated if they did pass it on?
None of these therapies should or will impact germline, and so there is definitely a need to be cognizant of that in the next generation and with family planning, all right? But as therapies come online, hopefully that you'll have options to manage that. But none of these, they will specifically exclude germline therapies.
Would they be available should they find their children?
Then you're getting into cases of pre-implantation genetics and so forth, right? Where you can do pre-implantation genetics to know, all right, is this a fertilized egg that we're going to want to develop into a baby? And that happens all the time right now, right? Where through in vitro fertilization, you'll actually, in processes, be able to test and say, I want to choose the healthiest eggs and implant. And that's fine. That happens all the time right now. But we're not talking about modifying those eggs. It's the natural process, simply doing a test before you implant healthy eggs into mom. Make sense? So we're not genetically manipulating eggs or sperm. We're just testing to see if they're healthy to move on. That's question, please.
Okay. I think all the things that you're talking about follow the FDA ruling of end-stage therapies, where you're only impacting people that are near death or will die relatively soon. When are we going to get to the point of being able to work with our population, which
Mhm.
and we take 12, 20, 30 years to get these approved.
So the question is that this all looks like end-of-life treatments, and when is it going to actually be helping a population that it's quality of life? And I would say we're there. Because with SMA, the FDA approved in adults, even though there weren't trial data on adults, they approved it in adults that are not going to die. Does that make sense? It's a bit of a stretch because still there are some pretty significant health issues, but I know some SMA folks, and these are some of the brightest folks. It's
I guess I want to hear that there's
hope.
patient lobbying groups or advocacy groups that are really pushing the FDA and pushing Congress for a population that is still smart, can still contribute if their bodies allow it.
He's looking for a voice to make sure that our voice is heard. And I would say our voice is heard, but we can't ever speak loud enough. We all need to advocate on our behalf to make sure that this is moving appropriately. And in a lot of cases, it comes down to money. We need to raise funds to make sure that these programs, these research programs that are going to become therapeutics, move along. But we also need the advocacy. We need to have the medical experts. We need to have the researchers. All of this together is going to make us a strong community that moves us forward and speaks with a loud voice. That's the way I see it. And it's not magical. We need to build it, put it together, and do that hard work. Okay, now I've gone through 2 examples, and I'm over time by 20 minutes. Okay, no, I'm just joking. I'm not going to bite into your lunch. It's maybe a little, but okay. Spark. So I mean, you saw this miracle yesterday, and I rank it actually 3rd, even though it may be your most important take-home is seeing this awesome kid able to navigate. And I stole this slide shamelessly yesterday from Stefan, and I'm really grateful to him for it. So this is a kiddo. I didn't show the previous. Let me see if I can hit play here. Oh, it's not available. So what Stefan showed is kind of the before, where, I mean, he can't, this is a maze. I think you can see that this is a maze that has arrows on it with raised black surfaces. So there's differences in color, in perception, and you have to be able, and there's like this big red stop sign, and I mean, he can't navigate it, right? You don't want to watch a kid that can't navigate. You know he can't. But again, coming to that outcome measure, you need to be able to measure. This company, the Spark, made this as an outcome measure. They said, look, demonstrated he can't walk through the maze. He can't see it. And then he literally navigates this maze. He's stumbling, but literally, I mean, it's miraculous. This is a woman that was treated who says within days, nobody could have expected that, right? I could see vibrant colors. I could even see the Philadelphia City Hall clock tower at night. I can see stars in the night sky. We're not talking about just huge, big blocks of white and dark, but literally being able to see. Now, and like I said down here, this is a form of blindness that impacts 1,000 people in the world, and they did this. And that's the same as with PLS. Drug companies are not ignoring this. It comes with a big price tag in many cases, but it's not being ignored. And it's a proof of concept for a lot of things that are going to follow. All right, I'm going to go through other things. So CRISPR. I mentioned CRISPR as a technology. Well, this is a company called CRISPR Therapeutics. And I talk with them quite a lot, actually, in my lab. They have, what you can see here, is a very healthy pipeline of drugs that are coming. Some of those things might be very recognizable to you. Maybe some aren't, but beta thalassemia and hemophilia, some of these are on their list of targets and quite far along even. You can see these pipelines published publicly. Nothing here isn't public. There's plenty that I can't talk about, but all of this stuff is public, and you can see it. And in some cases, I mean, a lot of these are publicly traded companies. You can even invest. You can talk to them, and we're hoping to as well. I wanted to show this slide just from last year. It's about Duchenne muscular dystrophy, and I mentioned a little bit earlier, so I'll go through pretty quickly. But I was like, wow, this is pretty cool, right? This really may pay off in the long run. And I kind of said it as, you know, I don't want to hype it too much, but this is awesome. And now we've got an update, just, you see this June 19th, and we were talking about this just yesterday. Positive news. I mean, it's working, and it's incredible, right? We weren't sure that we could hope for this. And one measure, I don't have an outcome measure here, I don't have data, but what I do have is the stock price of the company, and that's one way to measure how things are looking, and it's spiking. It's going way up. Sarepta is a pretty amazing company, and they've actually got an approved therapy. Let me go to the next. They've actually got an approved therapy in some countries. It was very controversial. A lot of people thought that the FDA approved it way too early, and so this is a whole different therapy down here in their pipeline that's having positive news. It's a whole different delivery mechanism and so forth. So it's pretty cool to see that this is a multi-pronged approach. There's a lot of working with Sarepta and this group with patient advocacy groups like ours who also are advocating with the FDA. So in the Cure SMA world, we've actually learned a lot from the DMD folks, the Duchenne's folks in this area. We borrow each other's brain power a lot. This is I talked about this a little bit earlier. This is the poster that you can see right outside. So this is, I would say, a really cool collaboration. We just funded, and there's matching funds that are privately raised by the folks sitting right at this table here, Karina and her daughter Maddie's foundation, and a lot of folks like Allison that are on that effort and involved. And we're hitching our horses together, and we have a lot of the same medical advisors too. And this is specifically developing some gene replacement therapy for SPG15. So it's moving, right? It's a place to start. It's important to move in these directions. And I'd encourage you to speak to them about their journey, read the poster outside, and if we've got questions, we'll keep addressing it. Questions? Question.
Well, they be using this in other diseases like Huntington's.
Oh, that's a great question. She asked if this is going to be usable in other diseases like Huntington's. There's several clinical trials in Huntington's. I moved from diagnostics into clinical trials using some of the same sequencing technologies, and one of the reasons I want to do that is I want to be part of clinical trials like that. And I'm working on a couple of Huntington's clinical trials that are using molecular mechanisms. Remember I mentioned ASO, the antisense oligo, using some of this from that same company that developed that technology. They're using it for Huntington's just by coincidence. And pretty darn effective in SMA. I'm hoping it's going to be effective for that population too. But it's still early days, but it's being worked on very diligently. Lots of, it's a bad one. Boy, that's a bad one. There's a lot of bad ones, yep. But that's a late onset. So one of the sick ironies of Huntington's is you'll have kids not knowing that you all have a death sentence, and you want to, it can be awful, right? We need therapies in that area, that's for sure. Other questions? You guys have, this crowd has amazing questions. I love it. And not just for me, I mean for other speakers that I've been hearing. What else? I've got a couple of questions that were posted up on the wall. What I didn't get to, and I actually excluded it, was stem cell therapies. Induced pluripotent stem cells. I've got two questions here that folks posted out on the board. And again, these were like 3 questions that might be for me. There's probably 300 questions, and I just saw Dr. Fink walk in, that were posted, and put your questions up there that he may need to address, but I might be able to address a few of these. And they're focused on, a couple of these are on stem cell therapy, and you'll notice that I didn't focus a lot on it. And I think that's representative of the state of the art right now. I think that stem cell therapies are an important research tool, but not today an important therapeutic tool. I think there's a lot of work left to do. It's mostly in the preclinical stage. There's some hype in other countries, and there's some folks that, you know, are, I think, paying attention to that hype and trying to do what you can for therapies. And I do not believe that that is recommended at this point by any of the healthcare professionals that are involved with our organization. And probably that's an understatement, but it's worth talking with your medical professional about. But you know, I can understand you don't want to do nothing, but at the same time, be very, very wary of too much hype. And I think in a lot of that area, there is a lot of hype. And another question. Do you think there's a possible cure with CRISPR, both HSP and PLS? And so I'll ask all of you, is there hope with CRISPR? Could be, right? Could be. We've got some good proofs of concept, a lot of work left to do, but it's looking increasingly positive. Don't want to hype it, but increasingly positive. Question?
There was a recent scare just a couple of weeks ago about how CRISPR causes cancer.
Yeah.
And then later scientists came out and said, well, a lot of things cause cancer.
Yeah.
To a degree. And so what is your feeling on all that?
So the question is, can CRISPR cause cancer? And one of the concerns of that, and I think we've covered it pretty well, is CRISPR can change your genome. And you want to be careful of off-target changes that you did not intend. You want to make edits that are specific to the region you're trying to focus on, but you can imagine anything can make mistakes, and that could make mistakes in other areas of your genome and screw up things that are working. And screwing up things that are working is the very definition of cancer. Our cells, when we're in an embryonic state, are made to grow as fast as possible. And later in life, we shut down those mechanisms. And cancer is often when you're accidentally turning those back on in specific ways. That's an overgeneralization, but it's one way to think about it, right? You're reverting to an embryonic state and grow when you need that growth controlled. So if you accidentally screw up those repressions and you turn it back on, that can cause cancer. And there was concern that CRISPR could activate those because it's a matter of frequency. If you're treating thousands of cells, some of those may become cancerous and that can become a problem. But I think that's less and less of a concern. It seems like there's less off target, and some of the recent CRISPR advances are specifically to avoid off target. And that seems to be getting better. And I've seen some of that data in my own lab. I can't show it because a lot of that's proprietary and hasn't been published yet. But there is less and less concern about that as we get better with those molecular tools and hone them. But that's an important concern. We got to be careful of that with the FDA, right? Got to wrap. If you have questions, happy to talk to you anytime. So thank you. Thank you.